Alright, welcome back to another lecture video, everybody. I have recorded this twice now, or attempted to, and gotten over an hour in before I noticed that it wasn't recording. So this is my third time recording this, and I am afraid it's not going to work. So I'm going to do this in little 20-minute stints. So we might have to have multiple parts to this video. So I apologize about that, but I don't want to waste another hour of my life. So let's do this. So in this chapter, we're going to be looking at carbohydrates. Carbohydrates are a major source of energy from our diet. They're made from the element carbon, hydrogen, and oxygen. We often call these saccharides, which means sugars. Saccharides. Yeah, and so you can see here several different sources of these saccharides. Bread, pasta, potatoes. All of these, you're going to see, contain starch, which we'll talk about a little later. It's a polysaccharide. The objective here in this section of the video is to go over the monosaccharides and to identify them as either aldoses or ketoses, and to use another indicator that will tell us how many carbon atoms are there. Now, carbohydrates form like glucose, for example. Glucose is the sugar that we like. We call it dextrose sometimes too. So glucose is produced by photosynthesis in plants. So plants take carbon dioxide from the air and water from the ground using sunlight, and they reduce the carbon dioxide into glucose, and they store it. Now, in living organisms like animals and things that eat plants, we oxidize glucose. So this would be the backwards reaction here, the respiration reaction. We oxidize glucose in the presence of oxygen back into carbon dioxide and water, releasing energy for us to power our bodies. It's kind of the reverse of the reaction, though the actual steps we'll see when we get to the later chapters aren't quite the same. I'm not sure if we talk about photosynthesis in this chapter. I know I teach another biochem course where we go over the light and dark cycles of photosynthesis, but that's where carbohydrates start. They start in plants. Now there's a few types of carbohydrates. There's the monosaccharides. That's what we're going to focus on here first. Monosaccharides are single sugar units. We know they're monosaccharides if they cannot hydrolyze. Hydrolyze means cutting with water. So a monosaccharide with water will do nothing. It'll just stay a monosaccharide. Now, what we know is that this bond, though, the hemiacetal bond, does hydrolyze. So water will actually cause our ring structure to open into a straight chain structure with the aldehyde group back on it. But that isn't becoming a simpler monosaccharide. That's the same monosaccharide. Now, a disaccharide has an acetal bond that links two sugars together. Now, these can be hydrolyzed. Monosaccharides are the product of a disaccharide hydrolysis. Molasses is an example of a disaccharide containing compound. It has maltose, and we'll go over disaccharides in a minute. We're going to go over the three most important monosaccharides here, though, first. Glucose and fructose are two examples of that. And then polysaccharides just mean many, so many monosaccharide units linked together. All of these acetal bonds here are linking these sugar molecules. And of course, hydrolysis will yield smaller chains, which we call dextrins. Those can be hydrolyzed into disaccharides, which can also be hydrolyzed into monosaccharides. So monosaccharides contain several hydroxyl groups, and you can see them there. Several hydroxyl groups attached to a chain of carbon atoms, either 3 to 8 carbons long for our sugars. Now, they can contain an aldehyde group, and if they do, we call those aldoses, or they contain a ketone group, and we call those ketoses. So erythrose is an aldose. It's a 4-carbon compound that has an aldehyde group, and you'll notice that all the other carbons have hydroxyls on them. And erythrulose is a ketose, also a 4-carbon sugar, hydroxyl groups on the other carbons, but the ketone group. So these differ on carbon number 1 and number 2. Otherwise, they look like they would be the same. So monosaccharides can be aldoses or ketoses. Now, we can also talk about how many carbons they have. If you have 3 carbons, we can call you a triose. 4 carbons, you can be a tetrose. 5 carbons, a pentose. 6 carbons, hexose. 7 heptose. 8 octose. So we can combine these together now. A 3-carbon aldehyde can be called an aldotriose. A 6-carbon ketone containing group can be called a ketohexose, and so on. So quick learning check. Label these as either an aldose, an aldose or a ketose, and then try to add tetrose, pentose, or hexose to that as well. All right. So hopefully you said aldohexose and ketopentose there. Let's talk about chiral molecules. Chiral molecules sort of mean handedness. Like you have a right hand and a left hand, right, that fit into the 2 gloves here. If a molecule has handedness, it can actually be considered and must be named as a different molecule because as we saw, there are things called isomers. These are similar to isomers, and we'll go over the differences here in a second. But we want to identify first what makes these kind of isomers possible, and that is the fact that some molecules can be chiral. So we want to identify that. So the objective here is to identify chiral and achiral, and also to use Fisher projections to identify the D and L enantiomers of monosaccharides. So if we take a look at stereoisomers here, we need to distinguish these kind of isomers from the ones we know already. What we know before is called structural isomers. These are isomers that have the same molecular formula, but different bonding arrangements. So C2H6O can be ethanol or dimethyl ether, right, depending on where we hook up the oxygen. C3H6O can be propanal, but it can also be propanone depending on where we have the carbonyl. So these are structural isomers. Stereoisomers are not hooked up different. They are bonded in the same sequence, but differ in the way that they're arranged in space. So when we talk about left and right hands, we know that they're chiral because, for one, chirality depends on superimposability. So let's just say we have a left hand here, and we look at its mirror image. So imagine seeing the mirror image, or as you can kind of see it here. Actually, this isn't the mirror image of the, well, yeah, this will work too. Let's look at the mirror image of the right hand instead. So, yeah, this is a bad example. Mirror images that are superimposable are actually the same molecule. So if you look here, the right hand, when you look at its mirror image, they look like they're the same shape, but here we see the top of the hand and here we see the palm. Those are not the same thing. There's different sides of this hand. So you can't just lay this hand on top of the other one and have them exactly match up. This is a better way of picturing it. The gloves. If this was a mirror right here and we were seeing the mirror image, we can't just lay these on top of each other and have them occupy the same space like we can with the cups here. If this was a mirror and this was the object and this is the mirror image, if we were to drag this over, they would actually line up perfectly and be the same. So the mirror image of this molecule would actually be the same molecule. Anything with a left or a right hand side we can tell is non-superimposable, but it's kind of abstract here. Let's look at actual molecules. So if we take a look at molecules, the definition of a chiral carbon is a carbon that has 4 unique groups. Anytime you have a carbon with 4 unique groups, we have the possibility of getting a mirror image that is non-superimposable. We also could get a non-superimposable non-mirror image. That'll happen as we start to get more and more carbons. Looking at just one carbon in our molecule, so the simplest carbon structure really, we can see that if you have 4 groups, and if you look at the mirror image, they look like they're the same. If we try to superimpose them, we see that they don't superimpose. If we drag this molecule over, we see that the chlorines and the bromines of one of these molecules is on the wrong side. If we simply attempt to turn that molecule around so that the chlorine and bromine are on the correct side, then we face another problem. The hydrogen, in this case, would still line up, but in one of the molecules down here, the iodine is coming forward. If we turn this molecule around, the other iodine would be pointing backwards. And so they still wouldn't match up perfectly, just like the thumbs on our gloves wouldn't match up. Even if we turned one of the gloves around, they would be on the wrong side of the molecule. So whenever we have a molecule, a carbon that has 4 different groups on it, that carbon is chiral, meaning it has a mirror image of itself that may not superimpose. Now, to look at an achiral carbon, meaning a carbon that is not chiral or doesn't have a handedness, we can see that this molecule here has 2 bromines on it. Its mirror image here, if we were to rotate one of these molecules around, we would actually get to a situation where the mirror image and the structure we were looking at can be superimposed on top of each other perfectly. And so that just goes and reinforces for us that this was never a handed molecule. This was always a molecule that we were just looking at differently. But just like where stereoisomer, or where our structural isomers might be cis or trans, and we had to give them special names, here, because we have a chiral carbon, we need to designate it as either being the right-hand version or the left-hand version. Now, it's a lot more complicated than that, but in this chapter, we're not going to go too far into it, or I should say in this course, we won't go that far into it. Okay, quick learning check. The carbon that's highlighted here in pink, say if it's chiral or achiral. Achiral because CH2OH and CH2OH are the same group. Chiral down here because each one of these groups is different. That means this molecule would have a mirror image. It would have a non-superposable mirror image. There would be a left-handed and a right-handed version to this molecule. All right, Fischer projections. Fischer projections are 2-dimensional representations. These are just the rules. The most oxidized group is at the top, meaning our aldehyde or ketone. Vertical lines represent bonds going backwards. Horizontal lines are bonds going forward. And intersections are just chiral carbons. So here's an example of how we turn some regular structures into some Fischer projections. Notice the differences. Here, bonds and dashes are going backwards, the wedges are going forward, and here we just have straight lines. So they just mean the same thing. Notice here that the most oxidized group is at the top. We still do that. All the intersections are chiral carbons, so that's why this carbon here is not shown with an intersection, because it has 2 H's on it. It's not chiral. But anyways, these are Fischer projections. Now this is a aldotriose. This is a 3-carbon aldehyde. And notice that we're designating one of them L-glyceraldehyde and one of them D-glyceraldehyde. Now this is because if we take chiral molecules and we put them in a sample and we shine light through them, the light comes out rotated. It gets what we call polarized. And whether or not a sample of a compound rotates the molecule rotates the light to the right or to the left determines if we call it L or D. And so that's kind of what this comes from. Now, or actually I should say R or S, rectus or sinister, right and left. That's the chemistry designation. For us in biology and kind of biochem, we use L and D, levorotary and dextrorotary, but we also have a simpler way of kind of naming them. We look at where the hydroxyl group is on the last chiral carbon from the top. And if it's on the left, we give it an L. And if it's on the right, we give it a D. It's that easy. So, if we take a look at these 2 designations of erythrose here, we see L-erythrose, which is an aldotetrose, has an OH on the left-hand side. Now this is the furthest hydroxyl on the furthest chiral carbon from the most oxidized group. So it's usually the bottom one. And a D isomer. So L-erythrose and D-erythrose. Now notice that L-erythrose is a mirror image of D-erythrose. The OHs are on the left here, the OHs are on the right here. there's two chiral carbons. Technically, there's also a situation where we could have mix matching. So we could have an OH and an OH on opposite sides. We could also have that switched. These are the other two stereoisomers. When their stereoisomers are mirror images of each other, we call them enantiomers. So, for example, having two chiral carbons gives us four different sugars. We have L-erythrose, we have D-erythrose, and then we have L, or D and L. This would have a different name. This wouldn't be erythrose anymore. Erythrose is just when the two OHs are on the same side. So this would have a different name of the sugar. I'm not really sure what sugar this would be called. But this would be the D version, and this would be the L version because of where the OHs are. These two stereoisomers are called diastereomers. Diastereomers, because they are not mirror images. So the mirror images are the enantiomers, and the non-mirror images are the diastereomers. Now these may be mirror images of each other, so they would be enantiomers of each other, but they would be diastereomers to these sugars. And likewise, these would be enantiomers of each other, but these two would be diastereomers. Just a little terminology for you. We're going to be focusing on enantiomers, so no need to worry about all the different combinations, but it does make sense to at least realize that every chiral carbon could have two positions. Chiral carbons are actually pretty important in biology. Most molecules in nature have mirror images, and often one stereoisomer has a different biological effect than the other one. One enantiomer of carvone, for example, produced by a spearmint plant, tastes like spearmint, smells like spearmint, whereas the mirror image made by the caraway plant has the odor and taste of caraway and rye bread. Our senses of smell and taste are actually really responsive to the chirality of molecules. So this difference in orientation in space is something that enzymes and taste buds and all kinds of receptors can actually identify and distinguish. And another thing, our amino acids that we use in our bodies are predominantly the L form, whereas the sugars that we use are the D form. So we don't use D amino acids and we don't digest L sugars. Our bodies are very specific. All right, quick learning check. So identify each of these as either being chiral or achiral. Pause it and give it a shot. All right, hopefully you said that this one was chiral, 4 unique groups, and this one was also chiral, 4 unique groups. All right, try another one. Indicate whether the following pairs of Fisher projections can or cannot be superimposed. The trick here is just identifying if they're chiral or not. If they're chiral, they can't be superimposed. So give it a shot. So these cannot be superimposed because they have 4 unique groups, whereas the one down here has 2 hydrogens. There is a way to rotate this molecule so that they can be superimposed. One more learning check. Identify the following Fisher projection, as either being a D or an L enantiomer. So again, you're looking for the chiral carbon furthest from the most oxidized carbon, and then deciding if it's on the right or the left. So that would be D. I lied. One more learning check. Draw the Fisher projection for this molecule. So that means converting all those wedges and dashes into straight lines. And that's it. You don't have to do the CHO at the top. Often when we're going to turn these into ring structures, when we turn these into ring structures, we are going to like to see our aldehyde group at the top so that we know where, we'll get to that. So just to, I guess what I'm saying is CHO is representation of the aldehyde group, but we often will see the aldehyde group sitting on top of the Fisher projection. They don't always show it as CHO. All right, and that is how you turn a wedge and dash model into a Fisher projection. Alright, so I'm going to take an opportunity here to stop this video, and then I will be starting on part 2 of this momentarily.